So I do think that one of the great examples of G proteins, which they will expect you to know about on the MCAT, either from your biology days, you should have seen some of this, and then also certainly from biochemistry. One of the great examples of G proteins in action is the epinephrine signal, or what we call the beta adrenergic pathway. So on the plasma membrane, which we know, of course, is a nice lipid bilayer, we have some lovely ability for transmembranous proteins. And we have the ability across a plasma membrane on the outside of the cell for a hormone to come in and bind to a receptor on the outside of the cell that then results in this signal being transduced, hence signal transduction, or being carried. The signal is carried basically through this transmembranous protein across the cell membrane to the inside of the cell without anything really having to actually cross directly in through a transporter. We have the binding of the epinephrine specific receptor, and as a result, we see a conformational shift in the epinephrine receptor, which results in the alpha subunit of the G protein, which is this green unit here. So just, I guess, to rewind a bit, the G protein in this context contains an alpha, a beta, and a gamma subunit. The beta gamma subunit will remain in one place, whereas the alpha subunit, upon the epinephrine receptor being bound, the epinephrine receptor will have a slight conformational shift that results in the GDP that has been bound to the alpha subunit will release, and GTP can come in, and we have an activation of the alpha subunit of the G protein. So the beta and gamma stay behind, and the alpha subunit of the G protein will move across and bind to this enzyme, which is a transmembranous protein called adenylyl cyclase. AC is how it's abbreviated, this blue protein. What adenylyl cyclase does then is it takes ATP as its substrate, and it converts ATP into cyclic AMP. Cyclic AMP, you should know, is a really important signaling molecule. And cyclic AMP, two of them will go on to activate one single pKa. So for every one pKa molecule, we require two cyclic AMPs in order to activate each pKa. Then once we have the pKa activated, it will go on and do a series of phosphorylations. Phosphorylation is a type of covalent modification. It's basically a way that we signal different enzymes, for example, to either be upregulated or downregulated. Unfortunately, we cannot make, I know it would make biochemistry students' lives and our lives as biochemists a lot simpler if we could just sort of make a universal statement that every time something is phosphorylated, it's upregulated, but that is not true. So unfortunately for us as biochemistry enthusiasts, we have to basically know which enzyme is phosphorylated and upregulated and which enzymes are phosphorylated to be downregulated. One of the ways I suggest you do this is by summarizing. If you draw diagrams in your notes, try next to the enzyme name, when you're drawing your diagrams, try putting a red P for phosphorylation when it's downregulating the enzyme, and a green P for green is go and red is stop, right? A green P for phosphorylation that is upregulating an enzyme. So you have to keep track of which enzymes are phosphorylated to be upregulated and downregulated. And I think that's an easy way to put that into your visual diagram when you're drawing out pathways in biochemistry, is just by having green and red as your colors, and then putting a P for phosphorylation. If you get into more complicated things like acetylation and uridylation, you can do the same thing, but using maybe AC for acetylation, U, or even UD for uridylation, and still using the green and the red pens. Just a suggestion. Okay. Interestingly, we have to think about this idea that these signals forever. So when epinephrine binds to its receptor, it's not going to bind irreversibly. It's going to bind and then be released, right? And this, by the same token, this alpha protein, this G alpha subunit of the alpha subunit of the G protein, if it's behaving properly, it should not be active forever, right? We wouldn't want to have one single epinephrine binding to its receptor cause the G alpha protein to be active forever. So there has to be a way that it turns itself off. So how does that happen? Well, this G alpha subunit, they're sort of summarizing, they're showing, okay, this is how it's activated. GTP will displace the GDP, activating the alpha subunit. But this is the coolest part of, I think, of the alpha subunit, is that it actually has, it says down here, GTP bounds to GS alpha is hydrolyzed by the protein's intrinsic GTPase. This subunit has within it an intrinsic, meaning within it, ATPase, which is an enzyme that, GTPase, not ATPase, GTPase, that will actually hydrolyze the GTP back to GDP, which will inactivate it. And once the GTP is no longer attached to the alpha subunit, then this alpha subunit will move back to associate with the beta and gamma subunits and wait until the next epinephrine binds. Does steps 1 through 4 have to undergo twice to move forward with the activation of pKa? No. And the reason that is depicted in this diagram. For every single epinephrine that is bound, we actually make 20 molecules of cyclic AMP. So for every G alpha subunit that moves over and attaches to, or binds, I should say, to adenylyl cyclase, we're making about 20 molecules of cyclic AMP. So we know, I said that for every pKa that we want to activate, we require 2. Yes, therefore 10, I see on the chat box, exactly. So there are 10 pKas that are going to be activated as a result of this cyclic AMP that's produced from one, in theory, one epinephrine receptor being bound. Now we know that in reality we have multiple epinephrine receptors, and this is happening, multiples of what this diagram is summarizing. But what they're trying to get at here is we're making about 20 for every single epinephrine molecule that's bound, and then as a result of pKa requiring 2 for every 1 pKa, we have sort of a decrease here by a half, but then we go up exponentially as we see this phosphorylation. This pKa is going to phosphorylate phosphorylase B kinase, which phosphorylates the glycogen phosphorylase B to glycogen phosphorylase A. The whole point of this is not necessarily that you necessarily need to know all these enzyme names, depending on what level of biochemistry you've had thus far. But basically, what do we have at the end game? Glycogen is broken down into glucose, and blood glucose is released. Why would we want that? Well, in our muscles, when we have epinephrine that's released, we want to, basically, it's the fight or flight response. I got to have my muscles at, peak ability, right? To, run, or if I have to punch someone, oh gosh, I hope not. But I don't know. I have to defend myself somehow, so that my muscles need that extra glucose, right? The muscles need the extra glucose at that moment. And so we want to have it harnessed as quickly as possible and as much as possible. And as you can see, as a result of this cascade, the signal transduction cascade of multiple reactions, we end up with about 10,000 molecules of glucose as a result of one single epinephrine receptor being bound. That's pretty amazing. So we've basically taken, to summarize, in a G protein, this is an example of a G protein pathway. There are many I could have chosen, but I have a feeling they're going to ask you something about G proteins. And so this is, again, a way that some kind of hormonal signal can be delivered outside the cell and then, released at some point. It's not going to be reversed. It will be reversible. But it's a way that it can bind to the outside of the cell, but still transmit the signal to the inside of the cell and have a result, a significant result, in fact, in this example. Also, it's important that the epinephrine receptor not always be on. So you see this, if we're thinking about the epinephrine receptor, somebody who has really high anxiety levels may have a lot of epinephrine that is being secreted. Well, at some point, the epinephrine has to be desensitized. It has to not react to a signal. It can't just keep reacting over and over and over again over many, many, many hours. Now, just think about how much energy that would take because then you can't always be releasing this much glycogen over 3 or 4 hours in a row of epinephrine signals. So we also have to have a way of turning that epinephrine receptor off if there happens to be tons and tons and tons of signals coming all at once over a long period of time. We wouldn't want that, because we would basically exhaust all of our energy responding to that epinephrine signal, and our body has to balance things out. So this is how that happens. So they're showing the binding of epinephrine, and they're showing then that GS beta gamma unit stays, right, while that alpha subunit continues on, which we already looked at. But in the meantime, then the GS beta gamma unit, which you thought did nothing, but in fact it does something, and we're going to look at it. The GS beta gamma unit will then recruit what an enzyme called beta arrestin kinase to the membrane, which is going to phosphorylate residue at the carboxyl terminus of this epinephrine receptor. So this little red kind of terminus, that's the carboxyl terminus. Beta arrestin kinase that is recruited to this area by the beta gamma subunit of the G protein results in phosphorylation of these two residues. As a result of those two residues being phosphorylated, a different protein called beta arrestin, or we call it BAR, will bind to those phosphorylated residues. And as a result of the binding to those phosphorylated residues, then it will initiate endocytosis of the epinephrine receptor into a vesicle within the cell. And then a different, once the beta arrestin is actually dissociated, then the epinephrine receptor will move back into the cell. So this is a way to kind of prevent this signal just from being kind of utilized, and to also save your body from exhausting tons and tons and tons of energy, again, over many, many hours if the epinephrine continues.